Okay, hello everyone. So again, I'm making a recording of one of the lectures. So this time I'm doing the week 15 lecture for PSY4050 Quantitative Research Methods. So as I've done in previous weeks, I'm going to make this available as supplementary material. Also gives me an idea of how, if I've got the timing of the material correct. So this week we are looking at MANOVA, Multivariate Analysis of Variance. Okay, and the aims are of what we're going to cover in this session are to extend the MANOVA model and include more than one dependent variable. Okay, so we're getting slightly more complex with design. And to enable students, you guys, to interpret the results for one-way independent groups ANOVA. Okay, so we're going to look at a design with one independent variable but multiple dependent variables. So different to what we've done previously. And then we're going to explore the options for post-hoc testing following a MANOVA where we might have got a significant result. Okay, so at the end of this week, so after the lecture and lab, you should be able to interpret the results of a one-way independent groups MANOVA and evaluate post-hoc tests following a MANOVA. And we're only going to focus in this week on like a one-way independent groups MANOVA. There are different, you can have a repeat measures design, you can have more than one independent variable with multiple dependent variables. We're just going to keep the design as relatively simple as possible, just for the sake of learning this stuff. But these things do generalize. There are more options for designs. All right, so MANOVA, or as I previously said, multivariate analysis of variance, is what we're covering. And I'm going to start with a little bit of a recap on ANOVA and factorial ANOVA. So as you guys will know, because we've covered it now by this stage, ANOVA is a univariate statistical method because it only has one dependent variable. There may be more than one independent variable, in which case we have a factorial design, and those are the types of things we've looked at so far on this module. So we've looked at one-way ANOVA, where we've got one independent variable, one dependent variable. So the independent variable has got different groups, different categories, and the dependent variable is measured on a continuous scale. We then went on and looked at factorial designs. So we've got multiple independent variables. So we've got more than one categorical independent variable, and we've still only got one dependent variable. So those were the ones we looked at, where we looked at interactions between our two categorical independent variables to see what things were going on. And our independent variables, as it says on that slide, has more than two levels, so they can have two levels, or they can have more. They can have three. We can have 3 conditions, 4 conditions, 5 conditions for each independent variable, as many as we like, really. Okay, so just some examples. If you recall, when we looked at one-way independent groups ANOVA, so we've got different participants in each of the levels of the independent variable. We've only got one independent variable, so it's a one-way independent groups design. This is kind of just to illustrate what it looked like, what such a design looked like. This isn't a particular question we looked at, but for example's sake, let's say we have one independent variable, and that's rank. So participants are either a constable or a sergeant or an inspector. 3 different ranks there, so 3 different levels of the independent variable. And then our dependent variable is some scale measure of their job satisfaction. And the question we might be interested in with a one-way ANOVA like this is there any difference between the 3 ranks in terms of their job satisfaction? So we'd look at the means of the 3 groups, see if they differ significantly. Okay, so that was the simplest design we looked at. Factorial design maybe could look something like this. So we've got one dependent variable here, which is alcohol consumption, but we've got more than one independent variable. So the first independent variable is sex, which has 2 groups, 2 categories, 2 levels, male, female. The second independent variable here is age group, and that's got 3 groups, or 3 categories, or 3 conditions, or 3 levels, whatever you want to call it. The first is under 20 group. The second is a group that's 20 to 30 years old, and the third is a group that's over 30 years of age. So we've got 2 categorical independent variables, and we're still looking at how how they interact with, or how the DV, which is a scale variable, amount of alcohol consumption changes as a result of these different categorical independent variables. And if you remember, when we have a factorial design such as this, this is probably the most straightforward factorial ANOVA design, 2 IVs and 1 DV. We've got a number of questions or hypotheses that we're testing. So in this particular case, the questions would be, is there any difference between men and women in the amount they drink? So that would be, is there a main effect of alcohol? A second could be, is there any difference between the 3 age groups in the amount they drink? So that would be the main effect of age. And the third there is there an interaction between sex and age in alcohol consumption? For example, is there a sex difference for older people but not for younger people? So that is the third hypothesis, which is there an interaction between our 2 variables and the way they influence or have an effect on alcohol consumption? So maybe we might find, if there was a significant interaction here, that maybe when people are older, males consume more alcohol than females, but in the younger age groups, there's no difference between male and female alcohol consumption, something like that we might find. So some things to note, that both of these univariate designs are testing for differences on 1 DV between 3 or more means. And remember also from when we learned about ANOVA that the type 1 error rate is controlled for by conducting the ANOVA. And if post hoc testing is required, so if we get a significant ANOVA result, we probably need to do post hoc tests, because the ANOVA only tells us that the highest and lowest means are different. And if we've got 3 or more means we're comparing, we need to look at if any of those are different from each other. So if post hoc testing is required, different methods exist to control for the family-wise error rate. So we looked at a few things like Tukey, SNK, the Bonferroni correction. They vary in how strict they are. Note also that you can have repeated measures designed, but we've not really covered those on this course. We don't have time. We've got kind of a long way to go in 10 weeks. So we've just kind of focused on these independent groups designs. Okay, so MANOVA, in contrast, tests for differences between 2 or more conditions on more than 1 dependent variable with 1 statistical test. So it controls for type 1 errors. It controls for this issue of multiple testing. Okay, so much like ANOVA is better than just doing multiple t-tests, because it gives us 1 statistical test that controls for type 1 error. MANOVA gives us the advantage over just doing lots of different ANOVA tests, because it provides us with 1 statistical test that controls for these type 1 errors as well. And it does this by combining the dependent variables and assessing if there are any significant differences between the conditions of the independent variable on the combined DV, this new sort of combined dependent variable, which you'll sometimes see referred to as the variate. Okay, and as it's noted on the bottom of this slide, so we could do separate univariate ANOVAs. So if we had 2 dependent variables and we were looking and we had 1 independent variable, we could do 1 ANOVA with the independent variable on the first dependent variable, and then another random ANOVA with the independent variable on the second dependent variable. But that increases the risk of a type 1 error rate. If we do MANOVA, it combines those DVs into 1 score, and we're not inflating our risk of committing a type 1 error. Okay, so there's an example on this slide. So it's building on the sort of one-way independent groups ANOVA, but we saw a few slides back. So what we've got here is a one-way independent groups MANOVA, and the design might look like this. So we've got more than 1 DV. We've got DV1 is job satisfaction, so again it's a scale variable still. DV2 is stress, also a scale variable, so it's measured on some scale. And the independent variable in this case is police rank. So constable, sergeant, or inspector. So we've got three levels, three conditions of the independent variable, and we've got two different DVs. And the question we're asking is, with a MANOVA, is there any difference between the three ranks in their job satisfaction and stress combined? Okay. So, some things to note. That is quite, although, you know, we're into an increasingly more complex type of design when we've got multiple dependent variables. This is probably as simple as we could make a MANOVA design. But there are a few things to keep in mind, and they're similar. I guess they are similar to the kind of the caution messages that would go along with a factorial ANOVA, and is that theoretically there is no limit to the number of dependent or independent variables you could have in your design. So you could put lots of DVs, you have lots of IVs, and you could also include covariates if you liked. But as with factorial ANOVA, the more complicated the design, the more complicated the analysis, and the more difficult it is to interpret the output, to interpret the results and actually understand what is going on. So you have to think carefully about which variables to include in your analysis. And a very important thing to keep in mind with MANOVA is that you need to be careful with dependent variables that are highly correlated. Okay, so if you have two dependent variables and they're highly correlated with each other, that's potentially an issue. Okay, so I mean, you could, the reason being is they're both accounting for variance in the correlation between the two. It means that those two variables have a lot of overlapping variance. Okay, you could probably just use one in the design and you'd find the same sort of results. Okay, so how MANOVA works. So I've decided to keep this as I've been limited in the amount of maths and formulas and calculations that I've presented to you here for MANOVA, because we are getting into, in terms of the calculations, we're getting into more complex territory because we're looking at multiplication with matrices. So not just multiplying single numbers and adding them up, we're actually multiplying matrices, which is linear algebra, which is quite complex and can get quickly confusing. So I've kept the mathematical side of this kind of lecture to a minimum. So MANOVA tests where the mean differences among groups on a combination of DVs are likely to have occurred by chance. Okay, so similar logic really to ANOVA or factorial ANOVA, but now we've combined our DVs. Well, we've got multiple DVs and we've combined them, and a new DV is created that maximizes group differences from the set of all DVs. Okay, and the new DV is a composite score. That means it's a linear combination of measured dependent variables, and it's combined to separate the three groups, our independent variable, as much as possible. Okay, so if you think about it like this, we've got a matrix with participants in each row, but instead of one DV score for each participant, we've now got multiple DV scores. And ANOVA is then performed on the newly created DV, essentially. And the goal of MANOVA is to discover whether behavior, as reflected by the DVs, is changed by manipulation of the IVs. So we've got our group, our categorical variable, maybe we're manipulating people into different groups, and then we're looking to see how scores on the DV change, but this time that DV is a combination of multiple things that have been measured. Okay, it's a composite measure. So the calculations for MANOVA, I've just put it in words essentially. Firstly, several DVs, several dependent variables, are measured for each participant. This produces a matrix of scores, subjects by DVs, rows by columns, okay, as I said previously. And these matrices of different scores are formed by subtracting each score from an appropriate mean. So the mean for that particular variable, okay? So we've got our DVs for each part, our scores on the DVs for each participant, and we're going to then create a new matrix of different scores by subtracting the difference between each participant's score and the mean on each of the dependent variables. Okay. That matrix of differences is then squared. Okay. And we then square the difference. We then take those squared differences and sum them, and this creates what's known as a sum of squares and cross products matrix. That's the thing that's formed. So similar to ANOVA, where we took each score, we looked out the difference of each score from the mean, we squared it, then we added them all together to get a sum of squares. This time it's a little bit more complex because we've got multiple scores, we've got a matrix of scores. So we're taking each score and working out its difference from the mean, and then we're taking that matrix of differences and squaring it, so multiplying the matrix by itself, yeah, and then what we're doing is summing them all together, and that gives us a sum of squares and cross products matrix. It's usually termed an S matrix, sometimes see it denoted as SSCP, sum of squares and cross products matrix. And then from this, determinants of matrices are found, and these determinants can be viewed as a measure of generalized variance for a matrix. Okay, so if you think back to the way ANOVA is calculated, once we've squared all our deviations, we add them together to get the sum squares. What we then do is divide by degrees of freedom, and that gives us our measure of variance. And in this case, we're doing a similar thing, and that gives us something that we call the determinant of the matrix. Okay, and then the ratios between the determinants of matrices provide tests of hypothesis about the effects of the IV on the composite DV. So think back to ANOVA or factorial ANOVA. So once we had our measures of variance for the total variance, for the group variance, for the error variance. If you remember, these were in ANOVA, these were called mean square. So the mean square for the groups, the mean square for the error. And we worked out our F ratio by taking the mean square for the groups and dividing it by the mean square for the error. And we do a similar sort of thing with MANOVA, but this time we take our determinant for the between groups matrix and divide it by the determinant for the error matrix. Okay. Okay, so checking assumptions. As with kind of ANOVA, factorial ANOVA, with multiple regression, with this type of multivariate test we're looking at here with MANOVA, there are several assumptions that we must check for MANOVA, and I'm not going to cover all of them, all the ones on this slide in detail. And if you want to find out more about each of these different assumptions, then if you read the Tabachnik and Fidel 2014 Using Multivariate Statistics textbook, it's an excellent textbook. They provide a very useful discussion of what these different assumptions are, how they can be checked, and what needs to be the case for them to be met, for those assumptions to be met. Some of these are quite difficult to assess. On the slide there, you can see we've got sample size and missing data. If we've got small samples and lots of missing data, that can be problematic for MANOVA. Independence of observations, univariate and multivariate outliers, something called multivariate normality, linearity, which means the variables can combine in a linear way. Homogeneity of variance covariance matrices, I've put that boxes M-test. Multicollinearity, collinearity and singularity. So that's the issue with DVs being highly correlated. You can link to this issue of multicollinearity. So we've basically got two DVs that measure the same thing because they're so highly correlated, or they account for the same variance. And homogeneity of regression slopes. Homogeneity of regression is an issue with something called Roy Bargeman's step-down analysis, which is kind of a means of post hoc testing, which I'll say more about shortly. But of these, there are three really, at a minimum, that we really need to meet the assumptions for. And those are the ones I'm going to kind of really focus on or speak about. If you want to know more about the others, like I say, have a look at the Tabachnik and Fidel textbook. So the three assumptions that we really need to meet are independence of observations. So multivariate normality and what's called homogeneity of variance covariance matrices, and we can test that with what's known as Box's M-test. So I'll say a bit more about each of these in turn. So firstly, independence of observations. This is the assumption that observations are independently sampled from the population, and there's no relationship between the observations in each group. Okay, so it's not the case that in the observations come from the same individuals in different groups or something like that, or that we've got a cluster of observations that result from some particular function of the population in some way. Okay, so it's if our data is sufficiently large and it's been sampled randomly, in many cases even if not deliberately randomly, our sample is what it is, and our data points are largely going to be independent. They're going to be drawn independently from the larger population, and that assumption will be met in such case. The second one is multivariate normality. So significance tests for MANOVA are based on what's known as the multivariate normal distribution. Okay, so this assumes that the sampling distribution of means in each cell of all linear combinations of the DVs are normally distributed. Okay, so it's similar to the assumption that in order for us to run our ANOVA test, the data needs to be normally distributed. Multivariate normality is kind of much more complex than that. So it means that we need multivariate normal distribution on all combinations of our variables. And there's no direct test for multivariate normality. There's no statistical test that we can do that will tell us whether or not our data is multivariate normally distributed. Okay, what people often do, or what's a common thing for people to do, is to test each variable for univariate normality. So you could plot it, you could get a histogram, plot it, look at the residuals, see if it's normally distributed, or you could even do tests of normality, statistical tests of normality. But that doesn't guarantee multivariate normality. So as a rule of thumb, Tabachnik and Fidel, I think this is information that comes from them. If you've only got a few DVs and we've got a sample size that's large enough such that the smallest cell has at least 20 participants in it, this should be enough to ensure the significance test is robust. Okay, and we can meet that assumption of multivariate normality. So with unequal numbers of participants in our different groups, and only a few dBs, a sample size of about 20 in the smallest cell should ensure a robust F statistic or robust MANOVA results. Okay, the third one, which is the one we're really going to focus on when we run these analyses using SPSS, is what's known as homogeneity of variance covariance matrices. And it's tested using something called Box's M test. And this is a multivariate generalization of the assumption of homogeneity of variance. So this assumption that each of the groups has roughly equal variance around the group mean, which we check when we're doing an independent groups ANOVA. This is the multivariate equivalent of it, and it's because we're dealing with matrices, it's got a much longer and more complex name, and it's called homogeneity of variance covariance matrices. Okay, and in MANOVA, the assumption is that the variance covariance matrices of the DVs for each group are sampled from the same variance covariance matrix, and that they can be pooled to create a single estimate of error. Okay, so quite complex. We don't need to get too bogged down in that though, because there is a test, Box's M test, and it's a test for equality of variance covariance matrices. There are issues with this test though. It's a very sensitive test, especially to departures from multivariate normality. It can give us erroneous results. If you don't have multivariate normality, and as I said before, that's difficult to know whether you do or don't, and you use Box's M test to test for homogeneity variance covariance matrices, you will probably get an incorrect result. Because this test is very sensitive to deviations of normality, we can adjust the level of alpha for Box's M test to make it stricter. Okay, so if our normal alpha level is 0.05, it's been suggested that because this test is sensitive and will detect, it will suggest there are significant differences between the variance covariance matrices when there are only trivial differences. We need to make the alpha level stricter so we don't find a significant result when it's not really an issue. Okay, so the suggestion is, the rule of thumb for Box's M is we adjust the level of significance down from 0.05 to 0.001. Okay, so if it's less than 0.001, then we would say we had a significant Box's M test and we couldn't assume equality of variance covariance matrices. But if it wasn't significant, then we're probably safe to assume equality of variance covariance matrices. So it works in a very similar way to Levine's test. It's just there are some issues with this test because it is quite a sensitive test. Okay, so those are assumptions. The key three there are independence of observations, multivariate normality, which is hard to assess, and homogeneity of covariance matrices. That's the one we'll focus on really when we run our MANOVA tests in lab classes. Okay, so selecting a significance test, and by this we mean which. MANOVA, when you run this test in various software, including SPSS, it produces a table of multivariate effects similar to an ANOVA table, with main effects for each independent variable and interaction effects between the independent variables. And for each of these, it basically gives us 4 statistics. Okay, so it gives us a choice of 4 different ANOVA tests, or MANOVA tests in this case, and we can choose which one to interpret, essentially. The 4 are, as you can see on this slide, PSY's trace, Wilks lambda, Hotelling's trace, and something called Roy's largest root. And as it says at the bottom of the slide, you run your test, it gives you your F ratios, and it gives you multiple F ratios for each test. So it gives you these 4. You have to pick which one you interpret. Okay, you have to choose which one to report. So here's an example from a print of a multivariate test from SPSS. And the question is, as you can see here, which do you choose? So the independent variable here, where we've got different groups, is age group, and we've got PSY trace, gives us an F value 4.738. We've got 12 and 1338 degrees of freedom, and P is less than 0.001. Wilks lambda gives us an F value of 4.879, 12 and 1175.005 degrees of freedom, and P is less than 0.001. Hotelling's trace is an F value of 4.999, 12 and 1328 degrees of freedom, P is less than 0.001. And then Roy's largest root, we've got an F value of 13.511, 4 and 446 degrees of freedom, so much lower degrees of freedom, P is less than 0.001. So what we've seen is we've got 4 F values, they're all statistically significant, so which is the one that you should choose? Some are more robust than others. So some rules for choosing, if this was your output and you've got this, you can see they're all statistically significant. There are some rules of thumb really for choosing between which one you should use in which situation. So if you have an IV that has only 2 levels, then you should use Hotelling's trace. And what you'll find is if you've got an IV with only 2 levels, all those F values, PSYs, Wilks, Hotelling's, and Roy's largest root will all be the same. And Hotelling's trace is the most commonly used criterion. It is essentially, Hotelling's trace is essentially a multivariate version of a t-test, essentially. Okay, but if you have more than 2 levels, so you've got 3 levels of the independent variable, 4 anymore, then the F values you get will differ, as we saw on this slide here. They're slightly, 3 of them are very close, and Roy's largest root is very different to the other 3, but they will differ. And in general, Wilks lambda is the most commonly reported criterion. So it's the criterion of choice when all the assumptions have been met. So you'd use Wilks lambda in most situations unless you've got this issue with the assumption of homogeneity of variance matrices being violated. So if we had a significant boxes M test, we wouldn't report Wilks lambda. We'd need to use one of the others, and the one to use is PSY's trace. That's the one that's considered to be most robust. So if we can't assume homogeneity of variance covariance matrices, then report PSY's trace, because that's considered to be the most robust. So if we've only got 2 levels of the independent variable, go for Hotelling's trace. If we've met all our assumptions, and certainly if we're happy that we can assume homogeneity of variance covariance matrices, use Wilks lambda. In most cases, when you've got 3 or more groups, where you see Wilks lambda reported. If you can't assume homogeneity of variance covariance matrices because you've got a significant boxes M test, then report PSY's trace. Never really seen Roy's largest root reported. Okay, so effect size, as with previous statistical tests, we've often looked to see what the practical significance of the results are if we get a statistically significant result, and different effect size measures exist. So we've looked at use of correlation, use of partial letter square with ANOVA and factorial ANOVA. The effect size in MANOVA is usually the most commonly reported one, is usually assessed with eta square, and this is the proportion of variance in the DV attributable to the effect, so attributable to the differences between the groups in the IV. And eta square is very similar It's calculated slightly differently to partial eta square. It's interpreted in a very similar way with different guidelines for what's a small, what's a medium, what's a large effect size. So it's usually 0.01 for small, 0.06 for medium, and 0.14 and above for a large effect size like with partial eta square. So if you run your test and you get results like we saw in the table on the previous couple of slides ago, so you get a significant MANOVA result. So you get a significant F statistic. You usually want to know which of the DVs are affected. So which of the DVs contribute significantly to the difference between the groups and the IV. It could be all of them, it could just be some of them. So this means follow-up tests, like post hoc tests that you need to do when you do an ANOVA. And there are two approaches to doing follow-up post hoc tests with MANOVA. These are conduct univariate ANOVAs, so univariate F tests on each dependent variable separately, or secondly, instead of doing that, we could do something called Roy-Bargman step-down analysis. Okay, so let's look at these in turn. If we run a MANOVA, we get a significant result. We then decide to see which of the DVs contribute to significant differences between the levels on the IV. We could just do these univariate ANOVA tests. So you would use these types of univariate ANOVAs if your DVs are uncorrelated, but they usually aren't. There's usually some sort of correlation between your DVs. But if there was a very small and insignificant correlation between your DVs, then you're probably safe to use this approach. You're just testing things using univariate ANOVAs. And when we do this, it's just multiple testing really. The effects of the independent variable or variables on each DV are assessed via a univariate ANOVA, i.e. conduct ANOVA on each DV separately. And because that's multiple testing, then we should adjust our alpha level to avoid committing type 1 errors. And we could use the Bonferroni correction where we take 0.05 or alpha significance level and divide it by how many tests we're doing. So if we had 2 DVs and we're doing 2 univariate ANOVAs, we could just divide that by 2. Okay, so this approach, let's see, here's some notes really on taking this approach using univariate ANOVAs. So Tabachnik and Fidel in their textbook, they note that this approach is the most common one for evaluating the contribution of DVs, but they don't particularly like it. Okay, so it is the most common. If you use SPSS, it gives you the univariate ANOVAs table as well automatically. So it is easy to just look at that to assess which ones were significant contributors to differences on the IV. But if you do that, it kind of raises the question of why did you bother doing a MANOVA in the first place? Why not just do separate ANOVAs, really? Some of the issues that Tabachnik and Fidel raise about taking this approach to kind of do your post-hoc follow-up tests following a significant MANOVA is that if your DVs are correlated, and they usually are, there's usually some sort of correlation, then the univariate tests are not going to be independent of each other. So if 2 DVs are correlated, then if one is significant, the other is also likely to be significant because they share variance. Okay, so if you've got 2 correlated DVs and you do a univariate ANOVA on the first DV and it turns out that you get a significant result, you're almost definitely going to get a significant result on the second one as well because they share a lot of variance. There's a huge overlap in variance. So when this happens, there's an inflation of the type 1 error rate. And as it says there, this kind of violates the spirit of MANOVA. You might as well have just done ANOVAs to start with and maybe used the bond for own correction in the first place. Okay, so the second approach to a kind of assessing the contribution of DVs is to use what's known as Roy-Bargman step-down analysis. And to do this, basically, you take your dependent variables and you order them in terms of priority. So like when you're doing hierarchical multiple regression. So you have to do this, essentially. This is a decision made by the researcher, which are the most important DVs, which should go in the model first, which second. Okay. And what you do essentially is you based on theory, based on what you know about the research, what your predictions are, your hypotheses, you take your highest priority DV and you would perform an ANOVA with that highest priority DV as the DV. So first DV would be then evaluated with an ANOVA, as it says on that slide. What you then do next, and this is the kind of step down, is another stage where you take your highest priority DV and you use it as a covariate, and then you use your second highest priority DV as the actual DV and run an ANCOVA on it. So the second DV is then evaluated using an ANCOVA with the first DV used as a covariate. So we're assessing the first DV using ANOVA, see whether it's makes a significant difference between the groups. What we're then doing in the second step of this analysis is we're taking that highest priority DV and using it as a covariate, and we're looking to see how much more variance is explained by the second DV once we control for the first DV. Okay, and then the third step would be if we had 3 DVs, for example. In this case, the third would be to take our third DV and evaluate that using ANCOVA, analysis of covariance, that is, with the first and second DVs, the higher priority DVs based on theory, as covariates. So what we've done with these 3 steps for such a model is we've kind of put the first DV into the model, run an ANOVA on it to see if the differences between the groups are significant on that DV. And then we've run an analysis of covariance using the second highest priority DV, but treating the first as a covariate, so we control for how much variance that explains. And we're asking the question in that second step: how much variance does the second DV account for when we control for the variance accounted for by the first DV? In the third step, we take the third priority, the third order DV, and we again do ANCOVA, and this time we ask how much variance is explained by that DV when we've controlled for the first and the second DV as covariates. Okay, and etc. etc. We could carry on depending on how many variables we had in this particular model. And as it says there, this is multiple testing, so you should use the von Paradigm procedure to adjust your alpha level. So here we're doing 3. If this was our step-down analysis, we're doing 3 comparisons, so we divide 0.05 by 3. Okay. So just to reiterate about the order of variables, that you should note that for Roy-Bargman, you choose the order that the DVs enter the analysis. So you must specify that order. This should be based on your theoretical understanding of the literature, of the research you're doing, and you should specify beforehand the order and have a good reason for it. As you know, when we looked at kind of statistical regression, when you change the order, that can often change the results and how much variance is accounted for by different variables or combinations of variables, and you can get a similar result here. So you specify the order, and you run the analysis based on that order. You don't fish around keep changing it until you find the result that you think is most interesting. You can get different results if you change the order, as the analysis will be addressing different research questions essentially. Okay, so that's it. In summary, what we've covered in this is that MANOVA is an analysis we use when we have categorical group data and we have more than one scale or continuous dependent variable. And it works a bit like ANOVA, but what it does is it takes our multiple dependent variables, a situation we've not investigated before, and it combines them together to give us a composite dependent variable, a combined dependent variable, and it looks for differences between groups based on that combined dependent variable, whilst controlling for type 1 error rate. And if you get a significant MANOVA result, a significant F ratio, you have to do some sort of post-hoc tests. And commonly used is to do multiple univariate F tests, univariate ANOVA tests, multiple univariate ANOVA tests with some sort of Bonferroni correction. Or alternatively, probably a much better way to do things is Roy-Bargman, what's known as Roy-Bargman step-down analysis. There are other approaches to investigate or to assess where the differences are that are driving a significant MANOVA result, but we're not going to cover them at this time. So, references, a couple of references for you there. The Andy Field textbook, chapter 17. Andy Field gives a good explanation of MANOVA. In terms of the post-hoc follow-up tests, he recommends using something called discriminant analysis, which is mathematically identical really to MANOVA, but it is the exact reverse. Where in MANOVA we're taking groups and then we're assessing differences between the groups based on a combined DV. In discriminant analysis, we're taking scores on a continuous scale and we're trying to separate them into groups. So it's kind of like doing MANOVA in reverse. I haven't discussed that as a type of follow-up analysis. I've stuck with univariate Fs and Roy-Bargman. So don't worry about that too much if you read the Andy Field textbook. If you read Tabachnik and Fidel, which I would highly recommend, they give a good discussion of MANOVA in chapter 7. Okay, so that's it. I'm going to stop recording and I will see you guys in class.